Well, now that we have learned about organic molecules, we're now going to see how living cells work. And so some of it will be the manufacture of some of these molecules we've been talking about. Some of it will be the breaking down of some of these molecules we've been talking about. So on to the cell. So in the introduction slides, what do you have in common with a mushroom? For the longest time, people thought mushrooms were basically plants. And it's understandable, they don't move around. And so you might say, well, moving as animal, not moving as plant. But believe it or not, mushrooms have more in common with us than they do with plants. So at the cellular level, there's actually quite a bit going on in mushrooms that is similar to our own cells. The cytoskeleton, this is a great stain showing the framework of microtubules and microfibers, etc., inside a cell. The blue represents the nucleus, and most of the time you would never see a representation like this. But selectively staining it, you can see, oh, this whole structure within the cell. And many of you enjoy caffeine. Well, that caffeine that gives coffee that fun kick is actually originally evolved as a way of protecting coffee plants from herbivores. We'll get into that more later. And again, without the cytoskeleton, your cells would collapse in on themselves. So if you took the beams out of a barn or rotted the beams away in a barn, the whole thing falls inward. Well, the same thing would happen if something broke down the cytoskeleton in your own cells. Now, antibiotics. Antibiotics, literally antibios, are drugs that disable or kill infectious bacteria. The goal of antibiotic treatment is to kill invading bacteria while doing no damage to the human host. Most antibiotics are so precise because they bind to structures that are found only in bacterial cells. So researchers continue to exploit the unique structures of bacterial cells to design and discover new antibiotics. Some little invading agents here. Now, organisms are all made of cells. They're either single-celled, like most prokaryotes and protists, or they're multicellular, such as plants, animals, and most of the fungi. Cell theory states that all living things are composed of cells, and that all cells come from earlier cells. So every cell in your body, and in every other living thing on the face of the earth, was formed by division of a previously living cell. Now, you might say, well, how do we look at these things that are so small? Well, there's things called micrographs, micro small graph, drawing or viewing. So what most of us have been using most of our lives is the light microscope, which is represented on the top. And that's great if you want to make a quick preparation, if you want to look at something that's alive, if you want to look at something that's under relatively low magnification. The lower two are types of electron micrograph. So instead of light passing through the specimen, bouncing off some mirrors and going through some lenses and then eventually hitting an eyeball and setting off the receptors there, you're actually putting electrons through the specimen. And you are not looking at the electrons that are flowing through the specimen. What's happening is the electrons go through the specimen, and then an image is captured of of the pattern from those electrons passing through. So let me go through these in turn. So again, here's a light micrograph. This is again, think of a photograph, right? So this would be a micrograph. This is an image of something quite small. And again, nice thing is that you can use this to view living or dead specimens. So this is a light micrograph of a protist called Paramecium. This is a scanning electron micrograph, or SEM, of another Paramecium. Now you'll notice that you're seeing all the surface features. You're not seeing the insides of the guy. Well, if you think of scanning, right, just as you scan the room looking for something, you're scanning the surface, so S for scanning, S for surface. You're looking at the surface features of something, but you can see it in much greater detail than you would see in a regular light microscope. However, the specimens must be killed and carefully prepared to do this. So this is not something you say, grab a slide and put a drop of water in a cover slip. No. It takes a lot more work to prepare these specimens for that sort of viewing. The other type of micrograph we're seeing here is transmission, right? Transmission, transmitting, passing through the specimen. So this TEM is going to give you a view of internal structures. And again, you can get much greater detail and much greater magnification, but the specimen has to be killed. The specimen has to be carefully prepared, so it's not something you can do as casually as we do using a light microscope. So all three instruments have their uses. People say, well, one's better than the other. For given uses, any one might be better than the other, but they're all useful in their own way. Now the next slide gets into size ranges. And most of us are pretty good at the upper end of this, right? 10 meters would be roughly 30 feet. Okay, human is generally between 1 and 2 meters, closer to 2 meters, so 5-6 feet. So there's your human height. Believe it or not, some nerve and muscle cells are actually almost a meter long. You have some nerve cells that run down, say, the length of your leg. You have some very long muscle cells. So there are some individual cells that are quite long, but they're also quite thin. Now, moving down to scale, so centimeters. Point of reference, 2.5 centimeters is an inch. Right, so 2.54 centimeters is 1 inch. So a chicken egg, by this scale, is, okay, it's going to be a couple of inches in size, and again, most of us are familiar with the size of a chicken egg. Move down to 1 centimeter, so again, this is, 2 out of 5, so a little less than 1/2 of an inch. Moving down scale further, 1 millimeter. Now in the metric system, it's all by tens. So 100 centimeters is 1 meter. 1,000 millimeters is 1 meter, right? So C for, so century, right? So centi, milli referring to like millennium, right? Referring to 1,000. So 1 centimeter is going to be, we use this as sort of a larger scale, but once you start getting to smaller stuff, you'd be putting a decimal point on the left-hand side and getting to smaller numbers. So hey, let's use a smaller unit of measure. So millimeters, as I said, 2.54 centimeters is an inch. That means it's 25.4 . Okay, so again, 25.4 millimeters would be an inch, 2.54 centimeters would be an inch. So the nice thing about the metric system is everything's multiples of 10, whereas in the English system or imperial system, you got all these 12s and 16s. It's not quite as consistent to the brain. Now, a micron, the letter mu is what looks like a little U with a tail in the front of it. Okay, a micron is a millionth of a meter. So centimeter is 100th, millimeter is 1,000th, and micron is a millionth. So once you start looking into things that are really only visible past the naked eye scale, you're usually starting to use the unit of measure of micron. So most plant and animal cells are in that range they give here of 10 to 100 microns in size. But once you start looking inside the cell, you're starting to look at things like the organelles, well, then you're obviously going to be seeing things that are smaller than the whole cell. So you're going downscale of relatively large organelles like the nucleus, then down to the actual size of individual bacterial cells, which are much smaller than eukaryote cells like plants and animals, and then down to somewhat smaller organelles like mitochondria. Now you're down to 1 micron. And we're really not going to spend a whole lot of time going into these size scales, so don't get totally stressed out with this. But the next unit down is nanometer. And so when you see NM, we've gone from micron to nanometer. So I will put up a little diagram that sort of walks through all the metric scales, and that should help with this. But the thing I want you to notice is on the right-hand side, there are some things you can kind of see with the naked eye, but you could get more detail seeing them with a microscope. So what might look like little dots swimming around in a dish, you could actually see properly with a light microscope. And there are points at which, again, depending on what you want to look at, the surface or the internal detail, or how you wanted to prepare the specimen, there are points at which the use of a light microscope overlaps the use of electron microscope. So there are, again, it isn't like light begins and ends and then their electron starts. There are actually points where using either of these tools overlaps. But once you start getting down to, say, 100 nanometers and smaller, then you're pretty much restricted to the electron microscope. So looking at really small bacteria, viruses, smaller organelles like ribosomes, individual protein molecules, individual lipid molecules, now you're getting into the land where, okay, you'd have to use an electron microscope, and past that point, past around 1 nanometer, we're sort of past the point of properly using them. They're working on ways of imaging things like individual atoms and very small molecules, but we're not quite there yet, at least on a commercial level. So again, this is the scale you can see with the naked eye, and then a little bit further than that where you start having to use a microscope to see much detail. And then getting into progressively smaller and smaller units. So, how do we organize life? It used to be there's a game called 20 questions, and the first question was often animal, mineral, or vegetable. In other words, alive, animal or vegetable, or non-alive mineral. So back in the day, you had two kingdoms, plants and animals. Nice and tidy, nice and easy, not anymore. We now classify all life into three major groups called domains. So this is above kingdom as far as organizational level. Organisms of the domains bacteria and archaea are composed of prokaryotic cells. You can almost think of pro like pre. The pre, thinking of before, kary, K-A-R-Y, refers to nucleus. So these are cells that don't have a nucleus. These are cells sort of before nucleus, as far as anatomy of the internal anatomy of a cell. Organisms of the domain eukarya, that kind of gives it away, are composed of eukaryotic cells. These would be the protists, the plants, the fungi, and the animals. EU, that prefix means true or real. So true nucleus versus before nucleus. So obviously the big distinguishing feature right off the top of your head would be, whether or not they have a nucleus. But there's other things as well. So they give you a nice little table here comparing the prokaryotic and eukaryotic cells. For one thing, prokaryotic cells were here first. They first evolved over 3.5 billion years ago. The eukaryotic cells did not come along until about almost 1.5 billion years later, right? So prokaryotic cells own the place. Then the eukaryotes showed up about 1.5 billion years later. Now, prokaryotic cells are found in bacteria and archaeans. Eukaryotic cells are found in protists, plants, fungi, and animals. Prokaryotic cells, much smaller, much simpler inside. When you get to eukaryotic cells, they're going to be larger, they're going to be more complex. You're going to see all sorts of structures inside. Prokaryotic cells, most have a cell wall. Some have capsules, fimbriae, or flagella as external appendages. Plant cells have cell walls. Fungi have cell walls, but they're not the same as those of a prokaryotic cell. Animal cells don't have a cell wall. They're surrounded by an extracellular matrix, basically a glue that sticks them together. Now, both prokaryotic cells and eukaryotic cells have a plasma membrane, so there are some things that they do have in common. There are no membrane-bound organelles in the prokaryotic cell because one of those is the nucleus, whereas in eukaryotic cells you do have membrane-bound organelles, again, like the nucleus, the endoplasmic reticulum, etc. Prokaryotic cells have a nucleoid region, so they have sort of an area within that cell containing a single circular chromosome. Eukaryotic cells have one or more linear chromosomes packed within a nucleus. And again, both have ribosomes, but the ribosomes are not exactly the same. So yes, they both have a structure to make proteins, assemble proteins. We'll get into this in more detail later. But they're not quite the same, and that's why some antibiotics actually target the ribosome structure of a bacterium, because it'll damage them but not damage the host cell. So a little more detail on our prokaryotic cells. So again, many of them they typically have a cell wall that gives structure to the cell. There's often a capsule, a sticky coating on the outside. Then there's business for attachment, fimbriae, or structures for locomotion, flagellae. And again, things that you see in both prokaryotic and eukaryotic cells are things like the plasma membrane. So there are some things that are shared. And this is just a transmission electron micrograph. So the electrons passing through, so you see the internal details of a particular of an individual prokaryotic cell. And again, if you look to the left-hand side, you'll see the magnification is 18,700x. So highly, highly magnified. Now let's get back to our eukaryotic cells. It's interesting, when you get to these generalized cells, they tend to just stick to animals and plants. We'll talk about fungi later on, but you'll notice that they kind of leave them out and they make it still a binary plant versus animal thing in this chapter. So you have sort of generic animal cell at the top and generic plant cell at the bottom. Now, they conveniently bracket. If you look to the right-hand side of that cell diagram, you'll see three things marked as not in animal cells: the central vacuole, the cell wall, and chloroplasts. So these are three features you see in plant cells, but not in animal cells. Cell walls, okay, that one's probably not too hard to imagine. Think of the cell walls like cellulose, that the little boxes that the cells are composed of. The central vacuole basically means a sac or a little bag. Now, there are vacuoles that form temporarily in many animal cells, but this is a little different. This is actually a central vacuole. It's in the middle of the cell, and it's basically a container. It can contain everything from just water to sugar solutions to pigments, but it is a persistent, permanent structure. It's not a temporary thing that comes and goes like you see some of the ones in animal cells. And of course, chloroplasts. Plants are photosynthetic, and so this is the organelle that traps sunlight and converts it into chemical energy. We got a whole chapter about that later on too. Now, as far as the animal cell, for some reason they didn't bracket the things that are not in plant cells. If you go to the upper right-hand corner of that animal cell, you'll see centrosome and lysosome. Those are two structures that you see in animal cells, but you don't see them in plant cells. Other than that, other than those three things with the plant cell and those two things with the animal cell, pretty much everything else is similar. Right? They both have cytoskeleton, they both have plasma membrane, they both have endoplasmic reticulum, they both have ribosomes, they both have mitochondria. And I'll bring this up again later on. Think about mitochondria. Both animal and plant cells have them. Sometimes people think, oh, plants have chloroplasts, animals have mitochondria. No, the plants have chloroplasts, so they make their own food, which they break down with the mitochondria. Animals have to eat the food that they're going to break down with the mitochondria. So they both have that tool to break down material for food energy. It's just one generates its own food and the other has to ingest it. Again, here's our generic animal cell. And for some reason, they didn't bracket off. But again, in the upper right-hand side, lysosome and centrosome are two features that you see in animal cells, but not in plant cells. Okay, here they conveniently bracket. What is not found in animal cells. Pretty much everything else is shared. Now, if I can get this to play.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Okay, then. Now we have a plant cell.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the 2 membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
All right, then. So, membrane structure, right? This is something that we see in prokaryotes and eukaryotes. Everybody's got a membrane, right? So the plasma membrane is what separates the living cell from its non-living surroundings. That plasma membrane and other membranes of the cell are composed mostly of phospholipids, arranged in a two-layer sheet called a phospholipid bilayer. So the neat thing about this is that it's a fluid mosaic. Fluid because the molecules can move freely past one another, and a mosaic because of the diversity of proteins in the membrane. One way I like to think of this is, say you have a bathtub, and you fill it with water. Let's say now you're looking at the outside of a cell. Say you throw a whole bunch of ping pong balls into that bathtub. Now the ping pong balls can move past each other. They're not going, they're not jumping in the air and diving down, but in that plane of the surface, they're moving around. Right? That's what these molecules do. They can slide around past each other. So that's what they mean by a fluid mosaic. If you think of a typical art mosaic, it's pieces of glass that have been glued into position, right? It's not like that. These molecules can move freely past one another. Now, there's also a diversity of proteins in the membrane. So say you toss some bath toys in there. Now, some bath toys just float on the surface. Some might penetrate down into the water. There's a variety of forms that bath toys can take. Well, there's a diversity of proteins in that membrane. So let me pop ahead to the closer picture. So outside of the cell, this would be looking down onto the water in the bathtub. And what you're seeing is a hydrophilic head. So those ping pong balls would be like the hydrophilic head at one end of the phospholipid. So that end is happy with water. So the outside of the cell, because the cells all live in a watery solution, and then the cytoplasm on the inside of the cell is another watery solution. So both those heads are perfectly happy being in contact with the water. But their tails, the two little tails coming off each of those molecules, are hydrophobic. So the very nature of these molecules means if you add this material to an aqueous solution, they will naturally form these membranes. Because the hydrophobic tails don't want anything to do with the water, the hydrophilic heads are just fine with the water, and they'll arrange like this naturally. Now, moving on to the bath toys, right? Now we have embedded in that membrane various proteins. And you can see that some proteins penetrate, go from the top all the way through into the cell. Some only go a little ways, like halfway through the membrane. So there's a variety of these, and it's going to come down to what their function is how what their shape is going to be, how they're going to be placed within the membrane. So phospholipid bilayer, and then embedded proteins, and that is your basic cell plasma membrane. Now, plant cells have a cell wall made of cellulose fibers, right? So think of this as like a box. The plant cell walls protect the cells, keep the cell shape, right, because it's a rigid little box, and they also keep cells from absorbing too much water. If an animal cell absorbs too much water, it bursts. Plant cells don't do that. Those rigid cell walls think of it as like a box. It keeps the contents from, bursting open. Now, animals don't have cell walls. And most hold together by a sticky coat called the extracellular matrix. So whereas plant cells are sort of built, sharing, cell wall components, and they're sort of built as little blocks. Think of like blocks in a wall. Animal cells are just sort of glued together. They don't have quite that same structure. In addition, the surface of most animal cells have cell junctions. And these are structures that connect cells together into tissues. So cells can function in a coordinated way. You see movement in animal cells in a way that you generally don't see in plant cells. So think of something like muscle cells, right? muscle cells are going to be working together in bundles called muscles, and to work collectively, they have to have some connections together to be able to work together. Now, a good question is, how was the first 21st century antibiotic discovered? Well, one problem, as you know from natural selection, antibiotic resistance is something that's a growing issue, right? We are selecting for microbes that have resistance to existing antibiotics. So we dose the antibiotics, the ones that survive have more offspring, and over time you wind up having a population of mostly resistant organisms. So we're trying to always find new antibiotics. And one technique one person did was to look at soil bacteria. And this person took individual soil bacteria and put them in their own little compartments, and then buried those devices in the original soil. So it was kind of neat. It was able to allow nutrients to pass into the compartments, but it kept other bacteria from getting into those compartments. And, oh, this is just shows you the little compartments that they were using in the soil, the research on the lower left-hand side of the slide. And the upper arrow, I'm going to go to the next. This just gives you the timeline of our development of antibiotics in recent years. Here again is the compartments. And again, things that we target would be things that are not shared with host cells. You know, if you think of human cells, we don't have the same type of ribosomes, we don't have cell walls, and our chromosome is not a single loop, it's you know, a bunch of linear strands. So, back to our soil bacteria. So the bacteria were next tested for their ability to kill a bacterium called Staphylococcus aureus, otherwise known as Staph aureus if you work in the medical fields. And that's associated with MRSA, methicillin-resistant Staphylococcus aureus. It's also associated with tuberculosis, right? So this is a bacterium we would like to find a way to kill, you know, easily. Well, they found that the bacterium most effective in killing Staph aureus was a new species that produced Teichobactin, which is now a new antibiotic. So it's going to be a while before you're going to see it, you know, on a shelf somewhere. They're going to have to do is go through a series of tests and eventually human clinical trials, like we're hearing about potential vaccines for COVID. But the fact is that by just studying bacteria in the natural environment, they were able to find a new chemical, a chemical we had not previously known of. Now the nucleus, so right now we're talking about a eukaryotic cell. The nucleus is the control center of the cell. Each gene is a stretch of DNA that stores the information necessary to produce a protein. So again, think of a gene as an individual recipe for a particular polypeptide. Now, those polypeptides do most of the actual work of the cell, like moving stuff around, carrying out the functions of the cell, enzymes, all these guys. So obviously, the code that determines the proteins is going to be important as to how a cell functions. Now, in eukaryotes, the nucleus is separated from the cytoplasm by that double membrane called the nuclear envelope. There are pores in the envelope, so that allows certain materials to pass between the nucleus and the surrounding cytoplasm. Within the nucleus, long DNA molecules and associated proteins form fibers called chromatin, and you saw this in the video a little bit back. Each long chromatin fiber makes up one chromosome. Now, you might sometimes see in a picture an illustration of a nucleus or a photomicrograph of a nucleus, little dots within the nucleus. The nucleolus, or plural nucleoli, are prominent structures within the nucleus, and that's where parts of the ribosomes are made. I'm just going to pop ahead to the next picture. So again, you see the protein and DNA together form the chromatin, the nuclear envelope surrounding the whole thing. This is basically like they took a triangle slice out of the whole thing. That's why you see those cut lines. And again, things pass into and out of the nucleus via those nuclear pores. And you can actually see in this photomicrograph, again, we're looking at transmission electron microscopy. We're looking inside a cell. Magnification's 8,800, so highly, highly magnified, but you can actually see the individual pores in the surface of the nuclear envelope. And in fact, we can really zoom in. Now we're at 12.5 thousand magnification, and you can see actual individual nuclear pores, which is pretty darn cool. Relationship between DNA, chromatin, and chromosome. We're going to be revisiting this later on, so you're going to have this come back to you many, many times. The DNA molecule wraps around proteins. They're called histone proteins. And again, it's kind of like wrapping thread around a spool. Helps keep it organized, and we'll talk about some more of the function later on. So that DNA coiled around the little proteins forms that strand. Okay, that's a chromatin fiber. Now you take that and you coil it. Okay, so now you have this small, you have this coil made of that strand. Well, now you take that coiled strand and you coil that, right? So you're progressively packing more and more material into these progressively wider coils. So ultimately, you could have DNA that's, let's say roughly 3 to 6 feet in length, fitting inside a single cell. How does that happen? Well, they're ridiculously coiled. They're super coiled. So it's a progressive series of coils made of coils until you finally have this illustrated here, a condensed structure called a chromosome. Now, ribosomes, whenever you see some, that refers to a body, right? So you'll see that sometimes come up. So when you see something some, it usually refers to a little body or a little structure. Ribosomes are responsible for protein synthesis. So these are the guys that are taking the information from the nucleus and using it to stick it together amino acids to make a polypeptide. So in eukaryotic cells, components of ribosomes are made in the nucleus. They exit through those pores into the cytoplasm, which is where they work. Now, some ribosomes make proteins that remain within the cytosol. They're going to be used within the cell. Other ribosomes make proteins that are going to be either built into those membranes, like I showed earlier on, or even secreted by the cell. Right? So they're going to be packaged in membranes and shipped out of the cell. And this is a sort of accurate model of a ribosome in the process of making a protein. Now, I've got animations already up on Blackboard for this, so I've come up with various ways you can see this, because this is something that it helps to see multiple ways of this working. But in essence, the ribosome is reading the messenger RNA, which is what carried the information out of the nucleus. And that the pattern of nucleotides is going to be what determines which amino acids are going to be getting stitched together. Now, some are free in the cytoplasm. Some ribosomes are stuck to the endoplasmic reticulum. So if you look in this illustration closely, you can see little tiny blue dots on the surface of what look like blue lines. Those are ribosomes attached to the endoplasmic reticulum. In fact, we specifically call that rough endoplasmic reticulum because it has ribosomes studding on it, like sticking beads onto the surface of a piece of cloth. So how does this work? Well, DNA is going to transfer its coded information to a molecule called messenger RNA, or mRNA. And that information is going to leave the nucleus in the form of that messenger RNA molecule. So that exits the nucleus through pores in the membrane and travels to the cytoplasm where it binds to a ribosome. One way to think about this, say you've got a grandmother and she's got the family cookbook. She's not going to let you borrow that cookbook. She's not going to let all the grandkids and everybody else borrow that cookbook. But you can copy a recipe and take that recipe and go make something. And that's essentially what you're doing. The messenger RNA is copying the recipes, the genes that are in the DNA. And that bit of information, that copying of that one gene, is going to be what goes out to a ribosome to build a polypeptide or build a protein. So the DNA itself does not exit the nucleus, but many copies of specific parts of it are being made and sent out into the cell for proteins to be made. So that messenger RNA exits the nucleus through pores in the nuclear envelope, and then travels into the cytoplasm where it binds to a ribosome. And that ribosome is going to move along the messenger RNA, translating that genetic message into a protein with a specific amino acid sequence. We're going to spend a lot of time going into this in more detail later on. So again, DNA, if a gene is activated, the DNA unwinds, and that gene is copied in the form of messenger RNA. So if I'm writing notes, if I'm speaking in English and you're taking notes in English, you're doing something called transcribing. If I'm speaking in English and you're taking your notes in English. So it's the same language, right? So trans, across, scribe, write. You're writing in the same language. So you're making a copy of the DNA in nucleic acid language. Now, that mRNA, once transcribed from the DNA, is going to exit and make its way to the ribosome. And again, we're going to get into this in more detail later, but just sort of walking through it quickly. It's going to make its way to the ribosome, and there you're going to have something called translation. So if I'm speaking in English and you're writing your notes in Spanish, right, you're changing languages. So that mRNA is translated from nucleic acid language into protein language. And there's going to be another type of RNA that helps with that called transfer RNA, tRNA, which you can also think of as sort of the translator RNA. Okay, so again, we're going to skip over these. These are you can use for your own reviewing. Okay, endomembrane system, manufacturing and distributing cellular products. Sometimes you hear the analogy of a cell as a factory, right? The boss is the DNA in the nucleus, right? The boss has his own office, and only certain people can move in and out of the office. It's kind of like that, right? The boss doesn't go walking around the shop floor telling people what to do. He sends out minions. He sends out assistants to go tell people what to do, and that's sort of what the function of mRNA is. Now, the manufacturing parts are you've got the ribosomes doing some manufacturing, but you also have the endomembrane system also manufacturing and distributing cellular products. So endo means inside, membrane means membrane. The endomembrane system in a cell consists of the nuclear envelope, the endoplasmic reticulum, which comes in both smooth and rough forms, the Golgi apparatus, lysosomes, and vacuoles. These membranous organelles are either physically connected or linked by vesicles, little sacs made of membrane. So basically a little bubble containing something can move through the cell and then empty its contents into another area. So let me skip over to the illustration just showing you that both have these endomembrane systems. So the ER, or endoplasmic reticulum, is one of the main manufacturing facilities. ER produces a wide variety of molecules. It is connected to the nuclear envelope, so it is sort of a continuum of envelope from nuclear envelope to ER to the Golgi to the outer membrane. And again, it's composed of interconnected rough and smooth ER, which have different structures and functions. So looking at this here, so again, you can see some of the ER there has little studs on it, right? Those are the ribosomes. And then you have some ER that is smooth. It doesn't have anything on it. Well, the rough in rough ER refers to ribosomes, right? So rough, R for rough, R for ribosome. The rough stands for ribosomes that are, again, like studying the outside of the membrane. So the ER also makes more membrane. Right? If you're pinching off little bubbles and sending things off, it's making the membrane that's going to be packaging this stuff. So you're replacing membrane through the ER. Ribosomes attached to the rough ER will produce proteins that will be basically, if you go back to that video, they basically are spooled into the growing ER membrane. So you see a ribosome on the outside, but it doesn't make the proteins going into the outside, it makes the proteins going into the inside. And so those proteins can then be pinched off in little membrane bubbles, transported to other organelles, and in some cases exported to the surface of the cell and outside of the cell. Now, some things manufactured by the rough ER are chemically modified and then packaged into transport vesicles, these are little sacs of membrane, that will just pinch off of the rough ER. And again, these can go to other parts of the cell, or they can even be, they're basically sent to other parts of the cell. So you see in this illustration here, you've got that ribosome spooling that new polypeptide within the endoplasmic reticulum. That can then go to the end of one of those folds and pinch right off, and now you've got this little vesicle, a little transport vesicle that can move that protein around elsewhere. The smooth ER lacks those surface ribosomes. It makes lipids, including steroids, and helps liver cells detoxify circulating drugs. So smooth, I always think of like smooth, like lipids are smooth, sort of a smooth surface. So smooth ER, lipids, including the steroids, and detoxification. Now, the Golgi apparatus works in partnership with the ER, receiving, refining, storing, and distributing chemical products of the cell. So this is like receiving and shipping versus the manufacturing part of the factory. So a checkpoint would be a nice way to sort of get an idea of where you stand is take those cellular structures in the checkpoint and then write them in the order they would be used in producing and secreting a protein. Good idea for practice. Okay, so again, this looks like a strange stack of flapjacks here is the Golgi apparatus. And on the left is, of course, a photomicrograph. And it would be receiving vesicles up at the top, and they would be passing through these layers and then eventually making it its way down to the bottom, and those would be vesicles forming to send away the product. So they receive at the top and they send out at the bottom. So you can see here sort of a cartoon version of that. Okay, skipping over that. There, lysosomes. Lys refers to splitting or breaking down. Some, again, small body. So a lysosome is a membrane-enclosed sac of digestive enzymes found in animal cells. Most plant cells don't have these. Enzymes in a lysosome can break down big molecules like proteins, polysaccharides, fats, and nucleic acids. They're also good for destroying harmful bacteria, engulfing and digesting parts of another organelle, like recycling parts of a worn-out organelle, and sculpting tissues during embryonic development. If you look at your hand, you have this, structure with the distinct fingers coming off of it, right? The fingers and the thumb. But that started out as a little webbed paddle. Well, then how do these fingers get separate? Lysosomes are part of embryonic development is lysosomes breaking down the soft tissues between the series of phalangeal bones and all that. There are also hereditary disorders called lysosomal storage diseases, which are actually tend to wind up being lethal in early childhood. So they are really critical to cell function and, with our species, to human health. So again, here's, two examples. A lysosome, let me just skip to the close-ups. A lysosome digesting food, so it ingests the food in a little bubble. So that would be a food vacuole coming in, and the lysosome comes along, fuses with the food vacuole, and digests the food inside that little pocket. Because if you think about it, if the food just came in and the lysosomes were going to digest them loose in the cytoplasm, what would happen? The cell would also be digesting itself. So you have to contain these sort of reactions in chambers. It's either a temporary food vacuole or something like a stomach we'll talk about later on. Also good for recycling. They'll break down damaged or just worn out organelles, and so parts can get reused later. And this is kind of neat. You can actually see vesicles containing damaged organelles and little fragments in there. So this is an example of a vesicle that's actually showing the recycling of damaged organelles.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and move through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself.
Okay. So again, vacuoles are large vesicles with a variety of functions. And again, when we talk about plants, that large central vacuole is a permanent big vesicle. But typically what we're seeing in animal cells are temporary ones, like food vacuoles budding inward from the plasma membrane. And some freshwater protists have contractile vacuoles that pump out the excess water that flows into them from the outside environment. It's kind of like a leaky boat, and you're bailing out the leaky boat. You can see it in this video coming up next. Right towards the center of the guy, and you saw that little swoop. See it swelling and contract one more time, and boop. So, and you can sort of scrub this back and forth to watch this over again to see it. I don't really have a pointer function here to show it to you. But it's kind of neat. It basically fills up with water and then spits it out across the membrane. This is nice because it gives you, let me click over. This is nice because it gives you a still picture of that. So if you look at the upper one, it looks like a sun there. It's a vacuole with all these little channels draining water into it. And then when it contracts, it just ejects the water out of the body, out of the cell rather. Now again, central vacuoles in plant cells, this is a permanent feature. So these are not things that are formed and go away. And they can store organic nutrients, absorb water, sometimes even contain pigments. When you see the color of leaves and flowers and whatnot, they can be involved with that, or even contain toxins to protect against herbivores. So this is a nice diagram. It sort of gives you the quick and dirty of the whole endomembrane system. So with the possible exception of the nuclear membrane. So it's got everybody but the nuclear membrane in there. Okay, energy, chloroplasts and mitochondria. One of the central themes of biology was the transformation of energy, right? How living systems take up energy, how it's converted from one form to another. And every time you transfer energy from one form to another, you lose a little as heat. So eventually all the energy that enters living systems is given off as heat. Now, two organelles act as cellular power stations, chloroplasts and mitochondria. And most of the living world actually runs on the energy provided by photosynthesis. Now, these are basically solar panels. They're photosynthesis, photo, light, synthesis, making. Photosynthesis is the conversion of light energy from the sun into the chemical energy of sugar and other organic molecules. We'll get into the next chapter, we'll talk about energy a lot more. Chloroplasts are unique to the photosynthetic cells of plants and algae. And again, these are the organelles that carry out photosynthesis. And so one of those themes I mentioned early on was the whole business of surface area. You can kind of guess by looking at this picture what surface area is actually carrying out the reaction. It's all those little stacked membranes that you see in the center there. They're referred to as thylakoids. And a stack of thylakoids is a granum. And we'll get into this in way more detail later in the semester. Actually, only a couple of chapters away. Now, mitochondria are found in almost all eukaryotic cells. These are the organelles in which cellular respiration takes place. So again, you're going to see these in photosynthetic organisms and organisms that consume their food. That's how it gets the food. Ultimately, it gets broken down into mitochondria. So these are what produce ATP from the energy of food molecules. So again, chemical energy in a food molecule into chemical energy of ATP. And ATP is really the direct energy source for most of what happens in a cell, most of the activity in a cell. So once again, remember, plant cells have both chloroplasts and mitochondria. Animal cells just have mitochondria. And once again, you can guess where the reaction's taking place. Look where all the folds are. The reaction takes place on that inner membrane, and all those folds increase the surface area, so the more surface area, the more reaction can take place. Now, one neat thing about chloroplasts and mitochondria, they contain their own DNA, which encodes some of their own proteins made by their own ribosomes. So each chloroplast and mitochondrion contains a single circular DNA chromosome that resembles a prokaryotic chromosome and can grow and then pinch, divide in two, reproducing themselves. You might say, well, where the heck did this come from? Well, there's evidence that mitochondria and chloroplasts evolved from ancient free-living prokaryotes that somehow managed to take up residence inside larger host prokaryotes. So this is sort of the evolution of organelles, if you will. This phenomenon where one species lives inside a host species is a particular kind of symbiosis. Symbiosis, sim with biosis, living. Symbiosis takes all sorts of forms. There's parasitism, predation, but this is a really unique one because over time, over the thousands of millions of years, mitochondria and chloroplasts basically became more and more dependent on that host karyote. So they eventually lost the features that would allow them to live independently, and they wound up developing into inseparable parts within a larger organism. And fun fact, the woman who worked most of this out was up at UMass, not that far from here. And she was Carl Sagan's first wife. So I laugh because I actually got a chance to meet and talk to her a couple of times. Lynn Margulis, neat woman. Okay, so again, popping over that. Cytoskeleton. So this is a network of protein fibers extending through the cytoplasm, and again, serves as both skeleton and muscles for the cell. Actually, not just making a support framework, but also involved with movement of materials inside the cell. So the skeleton, again, provides mechanical support to the cell, like parts inside a building, beams and struts and all that, and help maintain that cell shape. Now, there's several kinds of fibers that are made from different proteins. Microtubules, as you can guess, are micro, tiny tubules, tubes. These are little hollow tubes of protein. The other type of fibers, intermediate filaments and microfilaments, those are thinner, but they're solid. And the cytoskeleton provides anchorage and reinforcement for a lot of organelles in the cell. And some organelles will use a cytoskeleton for movement. So there's all sorts of functions that these come to. And it's kind of neat. You can see, let me click to the bigger one. You can see this special staining where you can see all those microtubules inside the cell, giving it shape and obviously providing tracks across which things can move. And if you've ever watched a live amoeba, you see the cells seem to flow along. It's actually building microtubules at the advancing end and literally sort of growing it forward. The tubules extend, the cytoplasm flows, and the whole thing moves forward. And on the back end, it's breaking down these tubules and reabsorbing those materials. So it's literally sort of flowing the cell by movement of cytoplasm and microtubules. Now, in some eukaryotic cells, microtubules are arranged into structures called flagella and cilia. These are external extensions of the cell that help in movement. The word flagellum literally translates from a word root for whip. So eukaryotic flagella propel cells through a whip-like motion. You usually see them singly, like in human sperm, but sometimes you can see pairs or groups of them in the outer surface of protists. Now, cilia, that's the plural, singular is cilium, are generally shorter and more numerous than flagella. And these guys move in a back and forth moment, like a rowing team rowing. So like you see oars rowing a boat, they tend to move in coordinated action. Now we typically think of these two structures as propelling protists through water. But you also can have cilia extending from non-moving cells. For example, on cells lining the human trachea, commonly called the windpipe, cilia sweep mucus with trapped debris out of the lungs. These are click over past the sperm cell to there. So it's neat. These are cilia on non-moving cells. Because if you think about it, your lungs would be a wonderful place for all sorts of nasty things to live in, and places where you don't want dust and other junk to clutter things up. So your body has mucus that traps all these things. Well, how do you get rid of the mucus? Well, the cilia sweep that mucus upward in a phrase I love, the mucus elevator. They sweep it up the throat until you clear your throat, right? You bring up that mucus, and depending on the situation, you swallow it or you expectorate it. Either way, it's not a problem to your lungs anymore.
Flagella, or singular flagellum, and cilia, or singular cilium, are microtubular structures that extend outside some cells and that assist in moving the cell or the cell's surroundings. Flagella are much longer than cilia. When a cell has cilia, their number is large, whereas a cell will have few flagella or a single flagellum. Many protists have cilia, and the sperm of many plants and animals have flagella. Flagella and cilia are assembled from tubulin subunits organized to form a circular arrangement of 9 pairs of microtubules attached to a central pair. Flagella and cilia bend to cause movement of either the cell or the surroundings. The bending uses energy derived from the hydrolysis of ATP.
Now, do not worry about the terms 9 in arms, microtubule doublets, and all that. I just want you to see how this works, so don't worry about seeing any of this slide or animation on a test. Here's an example.
The Euglena acus in this clip was treated with nickel sulfate.
Nickel ion paralyzes the flagellum. You can see the extreme shape changes of the cell, which clearly show the great flexibility of the pellicle or specialized cell membrane. You can see the paralyzed anterior flagellum at the left end of the cell throughout most of this clip. At the anterior end, you can see the reddish spot that functions as a light detector.
Paramecium moves by coordinated beating of its many short cilia. The cilia form longitudinal rows along the entire body of the cell and into the oral groove. The beating action of the cilia moves the paramecium forward and backward. It also produces a slow rotation of the cell as it moves through the water. The cilia in the oral groove propel food particles into the cell mouth, where the food can be ingested by phagocytosis. Credit. Graham R. Kent and Rebecca L. Turner, Smith College.
Okay, so because human sperm rely on flagella for movement, it's easy to see why something that would cause problems with the flagellum could lead to male infertility. Some men with a type of hereditary sterility also suffer from respiratory problems because there's a defect in the structure that is both common to the flagella of the sperm and the cilia found in the windpipe, found in the trachea. Now, within a human population, the persistent presence of a disease can provide a new basis for measuring individuals who are best suited for survival in the local environment. Because Bangladeshis have lived for so long in an environment that is teeming with cholera bacteria, one might expect that natural selection would favor individuals who have some resistance to that bacterium. And this is Vibrio cholerae. Recent studies of people from Bangladesh have revealed mutations in several genes that appear to confer an increased resistance to cholera. So this provides both insight into the recent evolutionary past of how humans and bacteriums have interacted, but also it might help us find some ways in which humans, across the entire population, could avoid, being infected with the cholera bacterium. So people may start finding proteins identified by those mutations, right? Mutations are random changes in genes and DNA. Well, if you can find what proteins that is coding for, then those might be used to create a new generation of antibiotics. And that's the end of the chapter.